Good morning and good afternoon, everyone. I want to thank you for joining today's webinar. This is Lauren Friedman from Alzheimer's Drug Discovery Foundation, and I want to welcome you to the first webinar in a series presented by ADDF ACCESS and Science Exchange. Today's speaker is Dr. Birgit Hutter-Paier, Director of Neuropharmacology at QPS in Austria, and she will be presenting on predictive disease models for CNS drug development. Next slide, please. So before we get started, I wanted to mention that Alzheimer's Drug Discovery Foundation, or abbreviated ADDF, does have funding opportunities for biomarker preclinical, clinical trials, and prevention programs for Alzheimer's and related dementias. And we fund both academic and biotech programs around the world, so there's no geographic restrictions. And in addition to funding, one of the things we do is provide resources to help accelerate drug discovery. Next slide, please. And one of these resources is called ADDF ACCESS, and this is a program of the Alzheimer's Drug Discovery Foundation and Science Exchange, and it's meant to connect researchers with resources and tools to move their programs along. And so this includes a curated network of service providers, many of them focused specifically on CNS diseases, with some vetting by ADDF. Through the website, there's standardized contracting across providers to ease project initiation. The website also offers 24/7 support from PhD level concierge specialists who will help you find the right provider and help you get quotes and help you contract. And one of the last components is a library of educational resources. These include some static guides that you can find on the website and webinars like we're having today. And at the end of, after today's webinar, we'll be posting the webinar on the Resource Center of Access. Before we move on, I just ask that you save any questions, and for the end, if you have any questions, just please type them into the questions tab, and we'll be doing the Q&A at the end of the presentation. So without further ado, I'd like to introduce today's speaker, Dr. Birgit Hutter-Paier.
Thank you, Laureen, for your introduction. And thanks also to the other organizers from ADDF and Science Exchange. And of course, welcome to our audience from my side as well. So please allow me to first, sorry, that was too fast. Please allow me to first introduce QPS to you. We are a mid-sized global company supporting our clients in all phases of the drug discovery process. QPS is a privately owned company and has sites on 3 continents. We are well known for our client commitment and scientific approach. QPS Neuropharmacology's main location is in Austria. And of course, we work closely together with the other sites within Europe and abroad. Here's an overview of QPS core competencies. Starting with the early developmental phases here with neuropharmacology, then DMPK, toxicology, dermal and transdermal research services, all of the before mentioned supported by bioanalysis and translational medicine, and finally clinical development. Our seamless integrated services can support you from drug discovery all the way through to regulatory submission. Today I will not cover each of these sites in more detail, but you can see we offer both breadth and depth. In your discovery and candidate selection stage, QPS is able to support you with in vitro and in vivo pharmacology studies in the area of neurodegeneration and orphan diseases, as well as ADME and tolerability studies. Later on, we offer animal toxicology studies supported by regulatory bioanalysis, immunogenicity, biomarkers, PK/PD modeling, all the way to clinical studies. Let's now switch to the topic of today's webinar and the challenges that you face when developing a drug, especially in the CNS area. Over the last few decades, thousands of interventions have been reported to mitigate pathological phenotypes or improve behavior in animal models of neurodegeneration. To date, very few of these findings have resulted in target validation in humans or successful translation to disease-modifying therapies. These are the reasons why an appropriate animal model, a good study design, a reliable partner, and an open communication of all results is mandatory. The inability of, let's go back to the right targets. The right targets is one of the first things you need to keep in mind, and finding the right in vitro and in vivo models and make sure that they present the same target. The inability of most of the animal models to completely recapitulate the human disease is a fact. And besides that, there is a distinct variability in animal models due to variations in breeding, colony maintenance, and other factors. If you consider that hundreds of different BLEC6 mouse lines are available, BLEC6, which is the main background line for most of the neurodegeneration models, you can imagine how hard it is to hold variability at bay. Consistency would be one way to do so, but lack of sufficient standards in design, conduct, analysis of studies, especially of animal trials, often reduces their translational value. And finally, there is a publication bias due to underreporting of negative results in the scientific literature. And that is a problem for all kinds of researchers, no matter if profit or non-profit. The first question now is when to use animal models in the drug discovery program. When considering taking a drug in vivo and test its efficacy, make sure you have positive and conclusive in vitro screening results. This is necessary to be able to select the most promising compound and get an idea about doses. To address toxic in vitro effects might help to foresee complications later on in vivo. Additionally, you should have information on the physicochemical properties of your drug to be able to optimize the formulation, to know active metabolites, to make sure your compound is druggable. This will help you also to make decisions on, for example, the application route. Make sure that you have reliable pharmacodynamic and pharmacokinetic data. It is elementary to know whether your drug is going to pass the blood-brain barrier, but also other drug transport processes should be understood. Possible side effects with ongoing treatment could be better anticipated with this kind of data, and parameters like treatment frequency, treatment duration could be narrowed down. At that time, please already think about acute versus chronic and preventive versus therapeutic treatment approaches and discuss your ideas with the CRO's expert. Make sure you have enough information to come to a definition about other relevant study parameters. Sex of the animals, for example, age preferences, the background stain, I mentioned the BLEC6 mice, will very likely influence the outcome of your study. You should be aware of that. And last but not least, take care to reserve enough budget. On the other hand, make sure that you get the scientific integrity and quality you expect from your CRO. Each CRO team member should have in-depth skills in one of the things I mentioned here, study management, animal maintenance, and so on and so on. These are key skills your external R&D people should bring in. Let's start now with one of the requirements I mentioned before, which are the positive and conclusive in vitro screening results. It is absolutely mandatory that you have seen effects in more than one in vitro assay. Some of these assays not only addressing efficacy, but if possible also in vitro toxicity. On this slide you can see that in 3 different assays, a cell-free assay, an assay in primary neurons, and in a cell line, a test compound was able to exhibit anti-aggregatory effects. For your information, in the cell culture experiments, middle and right, we only used non-toxic doses. Another requirement is to have at least some information on the properties of your drug before going into an in vivo proof of concept study. DMPK services perform all necessary studies not only for regulatory filing, but also to help make the right decisions for your first animal studies, like gathering blood-brain barrier penetration data, or determination of the treatment route and duration, as well as frequency. On this slide, you see the relevant assays to answer your questions regarding absorption, distribution, metabolism, and elimination of your drug. Experienced scientists can guide you in planning all necessary steps. Just ask. But having in vitro data and some information on ADME in place, how to choose the most appropriate animal model. The right choice of animal model to be used should be driven by the molecular target and the mode of action of your test compound and nothing else. Here's some ideas how to approach the topic. Take your time choosing the best fit animal model. It should mimic one or more human symptoms of the disease your compound targets. It should exhibit a phenotype reflecting the mode of action, inflammation, tau pathology. Make sure that you evaluate power analysis data and choose an appropriate group size for the experiments you are considering in a specific mouse model. By doing so, you will automatically narrow down to more appropriate animal models. But be aware, there is no perfect model out. Therefore, define what's the most important readout for you. Is it behavior, cytokine levels, plaque load, neuronal loss, 1 or 2 of that? And especially in a first step, in the focus on disease targets and pathways rather than the complete pathology. And finally, plan to demonstrate mode of action in more than one model. This will strengthen your overall data significantly, and it helps you in your decision to move forward to clinical trials. When considering an animal model, make sure it mimics one or more human symptoms of the disease. Here I have chosen 2 AD pathologies as example, namely plaques and cerebral amyloid angiopathy, CAA. CAA, besides occurring in the normal elderly and being a common cause for hemorrhagic stroke, can be seen at least in a mild form in nearly every AD brain. CAA appears to develop at approximately the same time base as senile plaques and other pathological changes in AD. The immunohistochemical results shown in this slide derive from human brain tissue provided by Newcastle Brain Tissue Resource on the left side and from brain tissue from one of our AD mouse models on the right side. The upper row shows the amyloid immunoreactive area in the isocortex of human tissue of healthy controls, left, of various Braak stages. as well as of mouse brain of increasing age. In both human and mouse, a clear progression can be seen. In the lower row, we quantified CAA in the isocortex of mice, in the right graph and image, which is also increasing over age. CAA in human tissue, shown on the left, is not yet quantified due to a very low number of blood vessels in the provided tissue, but data published from other groups indicate an increase of CAA with disease progression. And when you compare the CAA images from human and mouse, you can see how close the similarities are between the two species. When considering an animal model, we suggest to also make sure your model of interest exhibits the appropriate phenotype. If your compound aims to counteract brain inflammation, you need to choose a mouse model showing increasing neuroinflammation over age. The graphs in this slide derive from 5XFAD mice, a well-known Alzheimer's model. On the left, the level of various cytokines in 12-month-old females are shown. In the middle and right graph, the progression of activated microglia and astrocytes in the brains of 3, 6, and 9 months old mice compared to their 6 months old non-transgenic siblings are presented. Middle is activated microglia, right is astrocytosis. As you can see there, the biggest window in terms of progression is between 3 and 6 months. Therefore, we would suggest to treat animals in that age range, starting with 3 to 4 months and treat until 6 months or later if you want to evaluate behavior as well, to hopefully show a slowing down of the progression, at least in neuroinflammation due to your drug. Here I want to point to another hallmark of AD, namely tau pathology, which is the target of many new drugs. Let's focus on one specific tau species, phosphorylated athreonine 231. Graphs and images in this slide show results from an in vitro as well as an in vivo model and from human brain samples, which were again provided by Newcastle Brain Tissue Resource. As shown in the left graph, in a neuroblastoma cell line stably transfected with human mutated tau, a significant increase of tau phosphorylation at threonine 231 could be detected and measured. In a tau transgenic mouse model, tau hyperphosphorylation significantly increased over age at this residue, as shown in the middle graph and the corresponding images. And quantification of tau species in human transentorhinal brain tissue revealed a significant increase with increasing Bragg stage. With this slide, I also want to emphasize another point, which is a translational drug development approach from an in vivo towards from an in vitro model towards an in vivo model, whereby both, to a certain degree, reflect the human pathology and exhibit the appropriate target for your drug's mode of action. Focus on disease targets and pathways. The target in this example is pyroglu beta, since its appearance highly correlates with Alzheimer's disease. Therefore, models expressing and accumulating this truncated and toxic form of A-beta seem to be especially valuable for AD research. Results from human brain samples on the left show a clear progression of this A-beta species from early to late Bragg stages. In the two other graphs, we can see results from 2 mouse models showing increasing levels of this truncated amyloid over age, replicating the human pathology. The right image is from the TB 2.1 mice, and it's out of a publication from Alexandru. This is the only slide with non-AD models of neurodegeneration that I showed today, namely with the widely known R62 mice and a relatively new BAC-HD rat model, both being models of Huntington's disease. Data obtained with these two models demonstrate that they display a very similar phenotype when we consider their motor behavior. Not in terms of age, of course, but in terms of the progression. As shown, a positive effect here would mean that you interfere with the progression in one or the other model. But if you show a positive effect in both models, this might increase your chances for a successful clinical study significantly. It's for sure extremely challenging to design an efficient first-pass proof of concept study, especially in a stage where you know little about your compound. But here a few hints addressing some relevant points. Choose the animal model that fits best to your drug target and shows an appropriate and hopefully stable phenotype. You never know that. Choose a CRO with experience, able to show historic data sets with high quality standards, a list of publications, and ask for client referrals. Be aware the CRO scientists will temporarily be external R&D members in your drug development program and handle them in that way. Discuss with the CRO's experts how to design the best possible study. Age, sex, number of animals per group, treatment route and duration, as well as all readouts you might be interested in. Plan your study together with the CRO's principal investigators or experts, and based on all the information gathered to set up a detailed study plan. And upfront define milestones, deliverables, reporting requirements, timelines, and everything you are interested in and everything that you value in a collaboration. With the help of the CRO's experts, you will learn about the available historical data and of the specifics of an animal model. Therefore, ask for publications with data gathered at the CRO, for internal data not published, and discuss about your needs and concerns openly. The graphs in this slide show A-beta levels in the cortex of 5X FAD mice. Different age groups for both male and females are shown, and the progression of the specific pathology is obvious. With data sets like this, of course, addressing your drug mode of action, you are able to make decisions on when to start treatment in terms of age, how long to treat, which sex to choose, and many more. All the things I already mentioned before. But some models of AD, as the here mentioned, not only show changes in APP expression and in amyloid levels, but display other co-pathologies as shown in the next slide. These four images show the main and co-pathologies of a 9-month-old AD mouse model. This transgenic mice develop senile plaques in the cortex and hippocampus over age. These amyloid plaques show a typical composition as shown in the left upper image, with a thioflavin S positive beta sheet core, which is green, and amyloid surrounding this core, which is red. The plaque morphology is very similar to that seen in human brain tissue of many AD patients, and the number and the plaque size increases over age in both human and mouse. But besides plaques, these mice also show a robust phenotype in terms of inflammation, like GFAP positive astrocytes in green in the right upper image, shown here surrounding a blue plaque core. In the left lower photo, a high number of astrocytes in green and activated microglia in red, close to a plaque with blue counterstaining are shown. And additionally, in the right lower image, a plaque-rich area had been stained for tau protein phosphorylated at threonine 181, indicating tau pathology at least in slightly older animals like these with approximately 9 months. So discuss with your expert about the phenotype of your model of interest and its progression, and make sure the model closely mimics one or more human symptoms of the disease. Coming to the last slides, I want to address one of the biggest problems for us as a CRO, which is the lack of information about the compound to be investigated. But also the quantity is often an issue. Therefore, please provide a large enough batch for the entire study, and if possible, store a retention sample at your site. Specify exact storage conditions before, during, and after the study. Additionally, make sure to offer information about the stability of your compound. The more the CRO knows, the better. Deliver an exact protocol on how to solubilize, dilute, and apply your compound. And make sure all information about the specifics of your compound, good and bad, is available to the study director or PI and the study personnel in charge. And at the end of this webinar, allow me to summarize what I think are the most crucial points when you start and run an in vivo project in any disease area. Above all, that's communication, especially at the beginning of such a challenging developmental step. Because of that, I suggest to ask for information, publications, historic data, ethical approvals, and the time to have them in place if not already. For quality standards, SOPs, and reporting lines, request information on the experience of your project manager and the personnel involved performing the study. And ask if there is a possibility to visit the CRO and talk with the people there running the experiment. Of course, if that's possible for you as well. So from your side, please provide as much information about your compound as possible to project manager and study personnel, and discuss the study design and study protocol in detail, and add everything that is relevant for you to this document. And last but not least, do not change the study design of your study once the study started, if not absolutely needed. This could very likely weaken the study outcome, no matter how good and predictive the animal model is. Thanks.
Thank you, Dr. Hutter-Paier. That was an excellent, very informative presentation, and I'd like to welcome any questions the audience might have, and we actually have a few questions already, so I will get started, but while she's answering questions, please feel free to type more questions into your questions tab. First of all, we have a question about PET imaging. So could you comment on validated models for PET imaging of AD models? My experience is that, for example, for APPSWE mice, a popular model, gives a lot of PET signal in the cerebellum because there are a lot of amyloid plaques in the vascular system. But so a typical ratio nucleus to cerebellum doesn't show the plaques progress. So that's one thing you could address if you can. And then part two of that question is, some excellent PET models of AD are not commercial. For example, there's one proprietary model from Roche, and as they're not commercial, it's hard to believe they're validated. So could you recommend a model that's validated for PET imaging?
Unfortunately, I'm not a PET expert, okay. but I cannot answer regarding validated mouse model for PET imaging. But I think that the APPPS1 mice might be at least partially be useful. In a recent paper from Sufal et al. In just published with APPPS1 mice, they investigated a blood-brain barrier transporter with PET and followed also increase of plaques in the cerebellum and in other brain parts. And in cerebellum, they were not able to see that in the 50 days and 200 days group. So when you want to study progression of plaque pathology and possible drug effect, you would most likely go into mice with ages between 4 and 8 months. So you should be safe with this model. I think AD-PET in general is especially challenging, since the synthesis of PET tracers needs to be exceptionally standardized. And a small difference in specific activity immediately leads to a false negative result. So I think some of the problems might also contribute from that. One more comment, since I asked one of my colleagues in Austria, who is doing a lot of PET research, Thomas Wannig from the Austrian Institute of Technology. What they do is they always confirm their PET data immunohistochemically at the end of a study and try to correlate PET data with the histochemical data. So that's their way how to deal with that.
Thank you. We have another question, and it is around neural recording devices. Most neural recording devices currently available for freely moving mice record less than 16 channels. When studying the effect of compounds, this presents a limited view of how the drug may affect multiple brain structures. Do you think there is a need, and do you see a tendency for CROs implementing higher channel recording devices so you can observe a larger number of sites within the brain?
Yeah, I think in the future, of course, this will be one of the next steps. For now, I think it's for most of the company, they want to have very easy to interpret results. So the more channels you record, the more complicated everything gets. I think both has its relevance. On one hand, having simple results being easy to interpret, and on the other hand, to record all the different brain regions and structures. Yeah, I think the future will show us where we go in that regard.
Very exciting. Okay, we have one more question here. Is QPS using PET for their studies right today?
We are collaborating very closely with the Austrian Institute of Technology. So we have projects together, internal R&D projects, and we also refer clients that are not using our animal models to them. In this internal project, we are currently investigating cross-spreads between APP and tau mice and rats together, and we will hopefully have these results soon.
Great.
So we don't do it directly, but we work closely together with this group.
And so another question, have you done MRI to quantify brain volumes and neuronal loss on any of your AD models?
Yes, also together with the Austrian Institute of Technology and with the Medical University Graz Neurology Institute, we did that on APP Swedish London mice and also on APP rats. So we have some experience. We don't do it on our own, but we have ongoing and established collaborations.
Thank you. And do you have an AD model with primates, or might you be working with someone who is doing this? Our attendee asks, do you think this kind of model will be much better than the low-level animal models in addressing the real pathological changes in human AD?
We don't have an human-like a primate model. We don't work together with the group in that regard. I'm not sure if this will be much better. I think the combination of different models might make the difference. I'm not sure how well, how replicable these models are, how many animals you can use in this research. So there are certain limits to that. Of course, scientifically extremely interesting. And again, maybe the future will show us.
For sure. And we have another follow-up question here. How much neuronal loss do you see in your AD models?
In general, most of the AD models have very, very little neuronal loss, if at all. Some of the models have at least synaptic loss. The TBA 2.1 model that I showed you, that's the model with pyroglue beta, high levels of pyroglue beta and accumulation. This is a model where we can see neuronal loss clearly already with ages of 8 to 9 months, but still it is significant, but it's not something that is obviously visible. So you have to do quantification, do that very exact, and do it in the specific brain regions. But yeah, we have models, but most of the models don't show neuronal loss.
Thank you. All right. And what do you think about a viral vector approach to set up AD animal model?
Very interesting question. Very interesting question, because we do a lot of research with viral vectors. So we do that with tau. We do it. We try to establish a Parkinson model. A lot of things going on here on our end. We are close to finishing a large project where we try to overexpress tau in the brain, which was possible. We see a nice tau pattern in the brain. We see spreading. It's really a good histological images that we could share with you. But unfortunately, we don't see any behavioral changes. We don't see any other neurodegenerative signs. So we will work also in the future with that. We do a lot of internal R&D, but currently we have not seen an effect and made a useful model out of it.
All right. Okay, changing gears a little bit. How reliable is it to validate findings on post-mortem tissue from AD brains? Is there any quantification on how high the impact is at the transcriptomic or proteomic levels?
Food is something I think I could follow up when I ask one of the experts
That's
for
fair.
that.
That's fair.
Why we but you know why we use this human tissue is also to have a certain comparison between the human and the mouse tissue. So we need to make sure that what we see is not typical for the mouse only, that certain crow expressions are seen in both human and mouse, but for the more sophisticated answers to this question could be given by other people that I could rely later on.
All right. That brings us to the end of our time. I know there's several more questions, and I apologize if we didn't get to your questions, but I will follow up with the speaker and we will get your questions answered by email. So thank you so much for your enthusiastic response. Thanks to Dr. Hutter-Paier for speaking, and thanks especially to ADDF ACCESS for bringing us this presentation today. Stay tuned, and I'd hope to see you at a future ADDF ACCESS Science Exchange webinar. Thanks so much.